From about 20,000 to 10,000 years ago, the San people of the Kalahari Desert in Africa mastered the art of creating lethal hunting arrows by harvesting the larvae and pupae of leaf beetles belonging to the Diamphidia genus. To manufacture this biological weapon, hunters dug deep into the sand beneath commiphora trees during the dry season to collect the plump grubs, then meticulously crushed them using stone or bone implements inside an ostrich eggshell or wooden cup before mixing the crushed tissue with plant sap binding agents to coat their wooden arrow shafts. When deployed via bow and arrow, the active peptide Diamphidia attacks the prey's bloodstream by destroying red blood cell membranes, causing rapid red blood cell lysis, severe internal breakdown, and progressive exhaustion that forces the animal to collapse shortly after being struck. Around 8,000 to 5,000 years ago, indigenous tribes across North America and the Middle East developed potent biological combat tools by extracting venom from wild black widow spiders of the Latrodectus genus and desert scorpions. To prepare the substance, gatherers carefully captured the arachnids by hand using wooden tongs, manually compressed their venom glands or stingers to extract the clear liquid into a small horn or seashell container, and blended the raw venom with pine tree resin or wild honey to prevent rapid biological degradation before smearing it onto the tips of their arrows. When launched from a bow into a target, the venom unleashes massive amounts of neurotransmitters via alpha-latrotoxin, inducing agonizing muscle cramps, severe neural overstimulation, and intense tissue distress that incapacitates the victim. From about 6,000 to 4,000 years ago, hunter-gatherer tribes inhabiting the cold, temperate pine forests of northern Europe and North America discovered how to weaponize the lethal toxins of deathcap mushrooms such as Amanita phalloides. To manufacture the poison, the natives sliced up the fresh flesh and gill plates of the toxic fungi and used heavy stones to forcefully press and extract a cloudy, peptide-rich sap containing heat-stable amatoxins and phallotoxins, which they then carefully brushed onto wooden arrowheads before shade-drying them to preserve chemical integrity. When shot into an enemy or prey, these toxins stealthily destroy cellular protein synthesis, causing silent and excruciating internal organ failure, massive liver and kidney necrosis, and delayed fatal hemorrhaging days after the initial wound. Around 5,000 to 3,000 years ago, the indigenous tribes of the Amazon basin, including the Makushi and Ticuna peoples, invented a sophisticated paralyzing agent, known as Curare, by utilizing the bark and roots of forest vines from the Strychnos and Chondrodendron genera. To produce this dense concoction, they stripped the toxic vine bark, chopped it into fragments, and boiled it slowly in large clay pots for many hours, alongside various auxiliary plant juices, to concentrate the extract into a thick, dark brown, tar-like paste that was subsequently smeared onto blowgun darts or wooden arrow tips. When discharged through a blowgun or bow, the active alkaloids block acetylcholine receptors at the neuromuscular junction, immediately paralyzing the victim's respiratory muscles and causing swift asphyxiation while leaving the meat entirely safe for human consumption. About 4,000 years ago, nomadic tribes across Siberia, the alpine regions of Europe, and high-altitude northern Asia began utilizing the deadly alkaloids derived from the roots of Wolfsbane or Aconite plants belonging to the Aconitum genus. The manufacturing process required digging up the small, turnip-shaped taproots of the plant during autumn, when toxins concentrated at their peak, grinding them thoroughly with stone mortars, and simmering the mash over low heat for hours to concentrate it into a sticky, dark brown paste that required handlers to wear leather gloves due to its extreme dermal absorption risk. When applied to crossbow bolts or hunting spears, the active aconitine rapidly penetrates the skin or bloodstream, triggering profound neurological chaos, severe cardiac arrhythmias, extensive paresthesia, and fatal respiratory paralysis. Around 4,000 to 3,500 years ago, ancient coastal and riverine tribes across East Asia and Oceania unlocked the lethal potential of tetrodotoxin by harvesting the internal organs of pufferfish from the family Tetraodontidae. To prepare the poison, the fishers carefully harvested the highly toxic ovaries, liver, and skin, mashed the raw tissues together, and buried them in sealed clay jars with salt, or left them to undergo a crude natural anaerobic fermentation that concentrated the grayish, viscous biological fluid far beyond the potency of potassium cyanide. This potent liquid was then coated onto jagged spear blades or marine harpoons, used in coastal skirmishes and large aquatic hunts, where a slight puncture would introduce the toxin to induce complete motor paralysis and eventual suffocation while the victim remained fully conscious. Approximately 4,000 to 3,500 years ago, indigenous communities in East Africa and the Indian subcontinent utilized the toxic botanical properties of the oleander plant, Nerium oleander, and the yellow oleander, Theridia peruviana. To create the weaponized extract, gatherers harvested large quantities of fresh leaves and tender branches, crushed them intensely inside wooden mortars with a small amount of water to rupture plant cell walls, and boiled the green mash on low heat until it reduced into a thick, highly toxic dark green plant paste. This viscous preparation was thickly coated onto heavy hunting spears used for large game, where the cardiac glycosides quickly disrupted the victim's cardiac rhythm, causing severe ventricular fibrillation, heart failure, and circulatory collapse within minutes. Around 3,000 years ago, early nomadic pastoralists and indigenous hunters in South and Southeast Asia harnessed the lethal properties of the Strychnos Nux-Vomica tree by exploiting its alkaloid-rich seeds. The manufacturing method involved harvesting the ripe, hard-shelled orange-like fruits, extracting the flat, disc-shaped seeds, drying them completely, and pulverizing them into a fine powder using stone mills before cooking the powder with acidic plant juices to create a bitter, muddy, and intensely toxic paste. This paste was smeared onto the tips of hunting darts and javelins, which, upon piercing a target, released strychnine to block inhibitory neurotransmitters in the spinal cord, inducing violent, tetanic muscle spasms, severe asphyxiation, and rigid body contractions. About 3,000 years ago, indigenous tribes in the tropical forests of Southeast Asia, including early groups in Indonesia, Malaysia, and the Indochina region, developed powerful dart poisons from the sap of the Upas tree, Antiaris toxicaria, and related species. To gather the material, hunters cut deep V-shaped incisions into the massive tree trunks to harvest the milky white latex juice into bamboo tubes, then boiled or sun-dried the fluid extensively in open clay vessels until it thickened into a dark, highly adhesive black resin. This resin was applied to the tips of blowgun darts and wooden crossbow bolts, delivering potent cardiac glycosides that rapidly ruined blood circulation, caused localized tissue irritation, and brought down large forest animals within a short distance. Around 3,000 to 2,000 years ago, pre-Columbian indigenous cultures across the Pacific coast and tropical lowlands of South America utilized the skin secretions of giant cane toads, Rabo marinus. To process the toxin, the natives manually harvested the amphibians, gently stimulated or pressed their enlarged parotoid glands behind the eyes to release a milky white or pale yellow toxic fluid, and either sun-dried the secretion directly into a hard, brittle crust or boiled it alongside plant juices to create a sticky adhesive coating for their projectiles. When smeared onto blowgun darts or spear tips, the active bufotoxins and bufotenin surged into the target's bloodstream, inflicting acute vasoconstriction, violent heart palpitations, and sudden cardiac arrest. Approximately 2,000 to 3,000 years ago, the Choco and Embera indigenous peoples of northwestern Colombia mastered the extraction of Batrachotoxin from golden poison dart frogs, Philobates terribilis. To obtain the substance without touching the lethal skin directly, the hunters used large leaves or wooden sticks to capture the frogs, then placed them near a low, gentle fire or pierced them with a small green bamboo rod to induce severe panic and thermal stress, causing the frog's dorsal skin glands to instantly exude a thick, milky white foamy secretion. The hunters then wiped the tips of their blowgun darts directly across the frog's back, producing a projectile coated with a neurotoxin that instantly shut down voltage-gated sodium channels, causing immediate muscle paralysis, respiratory failure, and instant death. From 2,000 to 1,000 years ago, indigenous hill tribes and forest-dwelling communities across Southeast Asia, including groups residing in the central highlands of Vietnam, Laos, and Cambodia, manufactured organic poisons using the white latex sap of native wild trees, such as the blood dog trees, Antiaris and related Euphorbiaceae variants. The production process involved carving deep grooves into mature forest trees to bleed out the thick white sap into bamboo reservoirs, followed by open-air evaporation or slow simmering over wood fires until the material congealed into a thick, dark, pitch-like paste. This adhesive poison was heavily plastered onto the tips of heavy hunting arrows and trap stakes, ensuring that any animal or trespasser scratched by the blade suffered rapid systemic poisoning, localized tissue necrosis, and profound circulatory failure.